Okay, so now let's start with. Huh? Thank you, guys. Is it okay? It says stop, so that means it started. Everything is good. Again, I'm pausing. There you go. Yeah, that's good. Okay. Now, the question is about glucose metabolism. Okay, so let's start with glucose metabolism, then we'll go to fructose, and then we'll go to galactose. Okay. Glucose, two things can happen. As soon as you absorb glucose, we'll talk about that step first. As soon as you absorb glucose from intestine into blood, the next step is you have to use this glucose. You have to take him to cells. You know, like if it's circulating, let's say you order sugar because you want it in your coffee. If the sugar bag is outside, it's useless. You have to bring it in. Okay, so the first thing you want to do is bring glucose into the cells. When glucose comes into the cells, immediately should be converted to glucose 6-phosphate. Glucose 6-phosphate. This is the first step in all sugar metabolism. After you digest and absorb monosaccharides, the first thing is you have to do what? Phosphorylate them, trap them. Same thing will happen with fructose. Fructose will become fructose 6-phosphate and galactose 6-phosphate. Okay, so all three of them will be the same thing will be used for all three of them. The first step. You see, when you have glucose, as far as we know, glucose looks like this. Okay? Now, 1, 2, 3, 4, 5, 6. Where are we adding phosphate? At position 6. PO4 is the phosphate. So if you're adding phosphate at position 6, now if this is glucose, it'll be glucose 6-phosphate. If it's fructose, fructose 6-phosphate. If it's galactose, galactose 6-phosphate. All of them are 6-carbon sugars. Okay. Now look, the thing with phosphate is, if you remember the structure of phosphate, it's P negative negative. It's a highly charged molecule. And if you remember that day one we talked about cell membrane. What kind of molecule can cross the cell membrane and what kind of molecule cannot cross the cell membrane? Glucose is large, so it cannot cross the cell membrane. But now there is a glute transporter. There's transporters known as glute. G-L-U-T stands for glute or glucose transporters. It can allow glucose to pass through, but as soon as you have this bulky molecule, phosphate, it won't fit through. So the first thing you want to bring glucose into a cell is to trap it there. You want to trap glucose within the cell, fructose within the cell, and galactose within the cell. So the first step is always phosphorylation, which means you have to add ATP, and then you will get ADP, and the phosphate is added to what? To one of these monosaccharides. This is the first step. This is called the trapping. And how many carbon are in these sugars, monosaccharides? 6. So we'll call the enzyme hexo. And then an enzyme that adds a phosphate, what would you call it? Kinase. Hexokinase. Okay. So the first step in monosaccharide metabolism is always what? Hexokinase enzyme. And this is called the trapping of monosaccharides, trapping of glucose, trapping of galactose, trapping of fructose. You trap them inside the cell so they don't escape. Because the channel is not a pump, it's a channel. If they can come in, they can leave. But glucose is an essential molecule. Fructose and galactose, you want to keep them inside the cell. Trap them until you use them. Are we good with that? So please understand, even though I'm not giving the entire cycle, I'm going very slowly. This first step is very, very important. So hexokinase is able to convert all three of them into 6-phosphate glucose 6-phosphate, fructose 6-phosphate, galactose 6-phosphate. Yeah. Take your time.
So the purpose for that, does that end up making it hydrophilic? That's why you move inside the cell?
No, they're both hydrophilic. Sugar is hydrophilic regardless. It's not hydrophobic.
Okay, so
Yeah, it's nothing to do with it. It's just to make it large, bulky molecule so it cannot fit through the channel. The glucose transporter.
okay.
Let me put it this way. This is a cell. Now, glucose cannot go into a cell directly. It can't do that. It's not a small molecule. It's a large molecule. And it's charged. So it has a specific transporter known as GLUT. GLUT. Now, GLUT is a channel. It's not a pump. So if sugar can go in, if the concentration of sugar inside the cell increases, they can leak back. They can go in and out. I'll just draw one thing. It's an open door. There's no, like, it's not a revolving door in one direction. It's not an escalator that goes only in one direction. This is a door that opens in both sides. So wherever you have too much glucose, it will go to the opposite side. It's simple diffusion. Okay. Now the question is, when we bring glucose here, we don't want it to leave. We don't want it to leave the cell. So we have to trap it. Very good. So when we trap it, what do? We add a phosphate group at position 6. To fructose, we add a phosphate group at position 6. To galactose, we add a phosphate group at position 6. Each one of them requires what? ATP.
So it happens inside the cell?
Yeah. Of course. Once it goes inside the cell, hexokinase, yeah. Otherwise, remember, if you add a phosphate, that glucose cannot be fit through this channel. It won't. So it has to happen inside the cell. Okay. This is the first step. First step of all monosaccharide or metabolism. The first cellular step in carb metabolism is what? Hexokinase. Okay. Next thing you want to know is this. There are specific monosaccharide kinase. General one is what? Hexokinase. It will act on any 6-carbon sugar. Hexokinase will act on any 6-carbon sugar and will add a phosphate to it. But for glucose, it is glucokinase, a specific one. For fructose, it's fructokinase. And for galactose, it's galactokinase. Okay, so they have specific ones. They have the general ones. So you have the general kinase, which is able to convert all three of them. Okay, but if you have glucokinase, it can only do what? Convert glucose to glucose 6-phosphate. Fructose will only work for fructose. Galactose will only work, or galactokinase will work only for galactose. Fructokinase only for fructose. Galactokinase only for galactose. Glucokinase only for glucose. Hexokinase is found in all tissue. Hexokinase is found in all tissue. All tissue. All tissue. All tissue. Fructokinase and galactokinase also found in majority of tissue. Now when it comes to glucokinase, it's only found in liver and kidney. Memorize that for now. You'll understand its implication later on when I talk about gluconeogenesis. Why is that important? Glucokinase. So gluco, although I put an arrow next to this, glucokinase is only found where? In the liver. Okay, so let me just write down. Sometimes I don't write and I think I make people confused. So glucokinase. And then you have fructose. It's only found on in hepatocytes and renal tubular cells. Kidney cells and liver cells, that's all. Okay. Okay. So let's talk about one at a time now. One sugar at a time. Let's talk about glucose. Oh, sorry. This was good actually. You know what? I want to go as far as this. When we have glucose 6-phosphate, now it's trapped in the cell. You can use it. You can use this for what? To make ATP. You can store it as glycogen. Okay. You can use it to make NADPH. You can also use it to make triglyceride. You can also use it to make what? amino acids. NADPH, and then let's say in here, ribose sugar. What's ribose? Do you remember that? It's used for DNA and RNA synthesis. The sugar base of it. Very good. So you can do so much with this glucose 6-phosphate, because all the machinery to do such thing is located where? Inside the cell. But you have to bring the substrate, which is glucose, first into the cell. First priority is what? ATP. First priority is ATP. If you want to use, you eat to sustain your body's functionality. So the first priority should be what? Making ATP. So glucose 6-phosphate will be converted to ATP. That's the first priority. But let's say you got enough ATP. You got enough. And you still have extra glucose roaming around inside the blood, and it's being trapped. Triglycerides, which is converting glucose into fat and storing it. Now it's common sense though. When you have no energy, what are you going to do with glucose? Use it for energy. Let's say you have a lot of energy. What are you going to do with glucose? You don't waste it, you don't throw it away, you're going to store it. First thing you want to store it where? As glycogen, temporary stores. And if your glycogen stores are full, then what are you going to do? Adipocyte storage or triglyceride or lipid storage. I mean, that's common sense. All of you know that. Okay? So each step is a huge mechanism by itself, and that's our job, to learn these 5 steps. And we'll go over these 5 steps. This step we'll discuss it in the lipid part, but this step we'll talk about now. This step we'll talk about today and today. These in amino acid we have discussed it, and this we'll discuss in lipids. So I'm going to talk about these 3 steps. Vice versa is true as well. If you lack glucose in blood, that means you'll break down glycogen, hepatic glycogen. If you don't have glycogen, it's depleted and you're still starving, you're going to start breaking what? Protein. Further, you're going to eventually reach lipids. Okay, so vice versa is true as well. But let's talk about how do we make ATP from glucose. First thing. So now glucose will be converted to glucose 6-phosphate. Glucose 6-phosphate is converted to fructose 6-phosphate, and I'm going to ignore that step. You should remember that. It's in your book. Okay, which is reversible step. Let me just see if I won't have enough room, so I'll just escape that, and then it will be converted to fructose 1,6 biphosphate or bisphosphate. Okay. So first, fructose 1,6 bisphosphate. You should remember the mnemonic for them. Glucose is first converted to glucose 6-phosphate. Okay, and then to fructose 1,6 bisphosphate. Glucose 6-phosphate can spontaneously become fructose 6-phosphate, and then that's converted to fructose 1,6 bisphosphate. And fructose 1,6 bisphosphate is then glycerol aldehyde 3-phosphate.
This is just changing the hydroxyl groups on it also?
I'll tell you. Okay. And glycerol aldehyde, you have the picture in your book, don't you guys? Glycerol aldehyde 3-phosphate is then converted to 3-phosphoglycerate. 3-phosphoglycerate. And 3-phosphoglycerate is converted to phosphoenol pyruvate, and then eventually what are we going to get? Pyruvate. How many carbon does a glucose have? 6. So let me write in blue 6 carbon. Okay, let me tell you, pyruvate has 3 carbons. So going from a 6 carbon to 3 carbon, what did we just do? We broke glucose into 2 3-molecule sugars. And this process, because you're breaking down glucose, is known as glycolysis. Glycolysis. Give me a second. I will tell you. Glycolysis. Okay? Glycolysis. Glycolysis is the process of breaking down glucose into a smaller sugar. At least, first, if you have a sugar that's 6 carbon long, that's 3, and then you cut it in half. If you cut it in half, what are you going to get? Yeah, you will get 3 carbon sugars, 2 of them, right? Not 1, 2. Because we cut it in half over here. What's your question, Hamid?
What did 3PG stand for?
3-phosphoglycerate. 3-phosphoglycerate. Okay, 3-phosphoglycerate. Flip the page. Let me see if you. No, go. Keep going. I'll tell you which picture to. So I can see. No. Go backward. Yeah. A lot more. In the beginning. Go to the beginning of the book. Beginning. First 3 pages. Okay, now a little bit more. Sorry. Maybe 13. Keep going until you reach carbs. You went too far. There you go. One more. Backward. Yeah. Before this. One more. There. One more. There you go. What page is that?
11.
Page 11. You should have the picture on page 11. Okay. 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 Now, so all we want to do is we want to break glucose into a smaller molecule. That's all we want to do. And the process of breaking down glucose to pyruvate is known as glycolysis. Breaking down glucose. Lysis. What does lysis mean? Break down. And glyco means glucose. Or you can say sugar in that case. We can do the same thing with fructose and galactose. But they have to join this cycle somewhere in the middle. Okay? So the first step was what? Enzyme number 1. I'll draw here. 1 is hexokinase. Hexokinase consumes 1 ATP. It consumes 1 ATP. And it will donate its phosphate, and you will get glucose 6-phosphate. And then there's a second enzyme, number 2, which is known as phosphofructokinase. Phosphofructokinase, PFK1. Phosphofructokinase. Okay? Phosphofructokinase. See, in your book I put numbers so you don't have to cheat. Memorize it first and then flip on the back so that for each number there should be an enzyme. The answer is on the back of the page. Yeah.
Why the first step is hexokinase, not glucokinase?
Very good. It could be glucokinase, but where? Only? Yeah. Liver, kidney, and pancreas. Pancreas has it too. Okay. So in those areas, most likely glucokinase. But in the other tissue, it's hexokinase. So both could be at the same time. Doesn't matter. It's the same enzyme. The only difference is glucokinase is more specific for glucose. Hexokinase is a general enzyme.
Does it request like more energy or?
No, same.
If we have hexokinase, why we have specific qualities?
Okay. One of the reasons is that glucokinase is more specific for those tissue who deals with glucose metabolism, like pancreas, like liver. So it has a more, how should I say that, Vmax. It requires less glucose molecule to be active. Too much glucose molecule to be active. Hexokinase is even a small amount. So hexokinase is more efficient. Okay? Yeah. Alright, so look. Enzyme number 1. Enzyme number 1 is hexokinase or glucokinase, one of them. Most likely hexokinase. Enzyme number 2 is PFK1 or phosphofructokinase. Okay? The answers are on the back of the page, in that purple box. They're numbered appropriately. I only did that so that students, when they write on the same page, you just read it and you don't really memorize anything. So you look at that picture, you say 1. What could be 1? What could be 2? What could be 3? You should memorize it. Otherwise, it won't be helpful. So the whole idea of that was for that purpose. The answers are on the back. Now, both of them will use 1 ATP. Look, by donating a phosphate, it makes bonds more energetic rate or so forth. Okay, glyceraldehyde. Okay, that's the major one. You can also call glycerol 3-phosphate. Okay, now so we got two of these, right? So that means from now on we're getting 2 of each. I'll tell you at the end of the day when I go over this picture what do you have to memorize, what you don't have to memorize. Okay, we're more interested in the overall process of this. Okay, now glyceraldehyde 3-phosphate can be converted to 1,3-bisphosphoglycerate with the help of glyceraldehyde dehydrogenase. So enzyme number 3, right? Glyceraldehyde dehydrogenase. When it's dehydrogenase in general, all dehydrogenase use what? NAD. And this will take NAD and produce NADH. NADH. 7. So let me just put 7 then. Pyruvate kinase. 1,3-bisphosphoglycerokinase or phosphoglycerokinase is this one. What number is that in your book? I don't know. This enzyme. So I'll just put 6. Hopefully it's 6. So these guys will release ATP. At the end of the day, we produce 4 ATPs. And we use 2 ATPs. The net production is 2 ATPs. Okay? Because you have 2 1,3-bisphosphoglycerate and you have 2 phosphoenol pyruvate. If each of them will give you 1 ATP, that means there's 2 of each, so a total of 4 ATPs are produced. Okay? Let me make it simple. You have to know what steps first, and then I'll make it simple. What is the whole idea of this thing? Why do you have to memorize? Please know the enzyme pyruvate kinase. Enzyme pyruvate kinase. Make sure you memorize it. Pyruvate kinase. Make sure you memorize phosphofructokinase 1. Make sure you memorize hexokinase. Okay. If you know the additional enzyme, it would be a great idea. It would be perfect, especially if you memorize glyceraldehyde 3-phosphate dehydrogenase and phosphoglycerokinase. If you memorize these, it'd be amazing. It'd be amazing. But the one in red, please memorize these ones. These 3 are irreversible. The 3 irreversible steps of glycolysis you have to memorize. 3 irreversible steps of glycolysis. The first one is hexokinase. The second one is PFK1. The third one that's irreversible is what? Pyruvate kinase. Okay. I'm going to write it down. Don't worry. Yeah.
What do you mean by irreversible?
It's only one direction. Your car can only move forward, not backward.
So from pyruvate we can't get glucose?
With that enzyme, yeah.
Without
The same enzyme cannot do the both direction. Okay. When I say irreversible, we're talking about the enzyme, okay? Pyruvate kinase can only convert PEP, phosphoenol pyruvate, to pyruvate. Cannot do the reverse. PFK1 can only convert fructose 6-phosphate to fructose 1,6-bisphosphate. Cannot do the reverse. Hexokinase can only convert glucose to glucose 6-phosphate. Cannot do the reverse. Other enzymes that I mentioned in between, they can do both forward reaction and reverse reaction, the same enzyme.
But none of them are kinases, so they can't move fast forward or reverse.
Some kinase can do, yeah. Yeah. Okay. So irreversible step. Let me write down irreversible step. Hexokinase, phosphofructo, PFK1, and pyruvate or PK kinase, pyruvate kinase. These enzymes are irreversible. They can do the opposite of the reaction. For example, let's start with hexokinase. As far as we know, hexokinase converts glucose into glucose 6-phosphate. It can only do this. Cannot do the reverse. This is not possible. The same enzyme cannot do both directions, can only do one direction. So hexokinase can only do this in one direction. Now, fructose 6-phosphate is converted to fructose 1,6-bisphosphate with the help of PFK1. And again, it's irreversible step. The same enzyme cannot do the reverse. Yes, Sam?
Isn't that also the rate limiting step?
PFK1 is the rate limiting step, yeah.
Do we need to know that?
Yeah. And then you have phosphoenol pyruvate is converted to what? Pyruvate. You see, when I use the abbreviations just for me and for diagram, it's easy, but in your book the names are there. Okay, so you should know which abbreviation, what stands for what. It's already there. Just for simplicity we do that. Once you study something a lot, you will know the names fluently. You won't have any problem. Then you can use the these abbreviation. So enzyme, this one is pyruvate kinase. It can only do forward, cannot do the reverse of the same reaction. So for reverse, we need a different enzymes. Whenever an enzyme, the same enzyme can do both processes. For example, I'll show you one that's reversible. Glucose 6-phosphate can become fructose 6-phosphate. It's the same enzyme that can do both reaction, backward and forward. And that's known as glucose 6-phosphate isomerase, or I'll just say isomerase. Okay, mutase and isomerase. Generally, isomerase and mutase. Isomer is a mirror image of something. Okay, it's a mirror image of something. Remember, glucose, remember what I told you about glucose. If glucose has 6 OHs, if they're all pointing down, for fructose, at least one of them will be up. It's a mirror image, in a sense. So that's what isomerization. Isomerization, you don't change the chemical formula. It's the same number of carbons, the same number of hydrogens, the same number of oxygens. It's just the shape is changed in space. Okay? Me, I am still me. This is me, and this is me. If you take two of my pictures, I look different. But it's still me. So that's the only difference is whether I'm wearing a hat or not a hat, in a sense. You know, the overall things haven't changed. So that's what isomers do. It's a reversible reaction. Whenever the same enzyme can do the forward and backward reaction, they're reversible. Irreversible reaction means one enzyme can only do one directional. Can only produce, not go back and produce the reactants. Okay? So these 3 steps are irreversible, therefore they're extremely, extremely important. Very, very important. You have to memorize. You have to memorize. Okay? You have to memorize. Before we go further, because I want to talk about how we make ATP. You know, there's some molecules we should know them. Let's see how all of this will make, because I want to make sure that we understand how do we make ATP. Okay, first of all, NAD is nothing more than hydrogen carrier. You know how we talked about B12 was a methyl carrier? Tetrahydrofolate was a methyl carrier, right? So SAM was a methyl carrier, that's all. Now, NADH is nothing more than a hydrogen carrier. Okay. FAD is a hydrogen carrier. Okay. So that's 2 that will be used in this process. So that's one thing. They're nothing more than a hydrogen carrier. For example, there is a pump that makes ATP. Okay. I'll give you bits and pieces first, and then I'll give you the whole cycle. On the inner mitochondria, inner membrane of mitochondria, IM, there is a pump. And this pump is known as ATP synthase. ATP synthase. Okay, so this pump that I'm drawing right now is known as ATP synthase. ATP synthase. So this is the pump that makes ATP. If it makes ATP, should it be driven by ATP? No, because it will consume it. There you go. You know, when you want to make electricity, those of you who are aware of electricity, you know, most of our electricity comes from where? Michigan and Canada and those areas, because they have high water falls and things like that. So have you seen those dams? And you know how dams make electricity? There's a propeller, water falls on it, and it rotates, and the motor rotates, it produces electricity, like a generator. There you go. Yeah. This is assumed to be exactly same thing, but molecular level, believe it or not. It has a propeller component to it. This pump is driven by who? Or this ATP synthase is driven by hydrogen ions. Hydrogen ions. Hydrogen ions. Okay. Now, if you want to run in, why do we have to make a dam to make electricity? Why do we have to accumulate water? To create pressure. So it comes from high pressure to low pressure due to gravity. So you can propel. Here we must create a high concentration or pressure of what? Protons. Because proton is going to go against its concentration gradient. So this should be simple diffusion. Diffusion is free. Molecule always moving from high to low concentration is called diffusion, correct? Molecules moving from high to low concentration is called diffusion. So we somehow have to generate this high electropotential gradient. And this will drive this ATP synthase. In the process, all the ADPs that were produced by metabolism and AMP will be combined with phosphate to make ATP. So this is the most important enzyme of electron transport chain or mitochondria. Okay. Now, do you think glucose is rich in hydrogen molecules? Yeah. As far as I remember, you told me, I didn't tell you that, you told me 6 carbon, 12 hydrogens. There you go. What do we care for then? Hydrogens. So glucose is a good source of hydrogen. So we have to use it for energy. We don't care for its carbon. We don't care for its oxygen. What do we care for? Only hydrogen. Only hydrogen, okay? So we're going to focus on glycolysis and Krebs cycle and electron transport chain. How do we produce this hydrogen concentration? That's our purpose today. Number 1, who's the carrier of hydrogen? Very good. They are simply a carrier of hydrogen. I'll circulate it. They just deliver them to this area. That's all. They're nothing more than carriers of hydrogen. They have no other role in that sense. Okay? So if you understand what's the purpose, it will make our life easier. Fatty acids are even larger sources of hydrogen. Fatty acid, usually the one we use in our body, is 16 carbon long. And how many hydrogens would they have?
46.
A lot more, because they don't have OHs. They only have H.
46.
Yeah. Okay. So they will have a lot more hydrogen. They'll produce 3 times more energy. So 1 mole of glucose or carbs give you about, I think about 4.5 calories. 1 mole of protein or amino acid gives you 4.5 calories. So it means the amount of energy you get from carbs and protein is the same. But lipids gives you 9 calories from 1 mole. Twice as much. Twice as much. Okay? So they're high energy sources. Okay. But as long as you understand this and this, we will be okay. Now, why do we have to break down to grab the hydrogen from it? If you don't break it, how do we grab hydrogen from it? We can't. Let's take a quick break and I'll come back to how do we make ATPs, okay?